in biochemical engineering that we are doing, and today we start with a new chapter which is called microbial growth. But before we start with the chapter, let's try and understand that what we have been doing and how this translates or how this flows into what we are trying to do in the next few classes. So we have been looking at the concept of enzymes, and enzyme immobilized and immobilized forms, and how that affects the process or catalyzes the process. How does it relate to is that in in biochemical systems, enzymes catalyze the process and so on, but many of the processes that are involved, or some of the processes at least that are involved, are growth processes. For example, the cell growth process. And what we are going to look at today and in the next couple of lectures is essentially the cell growth process. Okay, so let's start with the screen, and what you have over here is microbial growth. Now, what is microbial growth or cell growth in general? There's something called the cell cycle, and cell cycle is essentially the various events that occur during the growth of a single cell from its inception till its time of division into daughter cells, and that whole thing is referred to as cell cycle. So from the point where a single cell inception occurs to the time when it divides into daughter cell, that is known as one cell cycle. And there are several parts of the cell cycle, and let's try and understand what these different parts of the cell cycle are. So the first part is known as M phase. Okay, well, it's called M phase because this is where nuclear cell division or mitosis occurs, and that's why it's called M phase. So this is when the mitosis occurs. Next is interphase. So after mitosis occurs, daughter cells are formed from cell division, that is mitosis, and so two daughter cells are formed, and the cells enter into G phase or the growth phase. So you start with the M phase of the mitosis phase, then you go to what is known as the interphase, which is the phase between the M phase of the mitosis phase and the growth phase, or the G phase. Okay, then you move on to what is known as so after the interphase you move on to what is known as the G phase. Now, G phase, there are two G phases. One is called the G1 phase, and then there is another one we'll come to in a minute, which is known as the G2 phase. Now, what happens in the G1 phase is there is a high rate of biosynthesis. So essentially, once the daughter cells are formed, so there is growth of these daughter cells. So it's like a baby being born, and you give nourishment, nutrition to the baby so that the baby grows into a fully mature adult. So that is what it happens. So the adult cells, mother cells, generate the daughter cells, which now need to be replenished through growth, and therefore there is a high rate of biosynthesis, and this phase is known as the G1 phase. The next phase is known as S phase. So because what happens in this phase is DNA synthesis occurs. So the first G1 phase, essentially this is all part of the growth phase again, but the first G1 phase that we talked about is essentially biosynthesis of different elements, and these different elements come into the cell. And the next S phase is a very important cell where DNA synthesis occurs till the DNA content of the cell has doubled. Okay, so that's the DNA synthesis occurs until the DNA content of the cell doubles. And then is the G2 phase, the final phase, which is the initiation of the mitosis. So what happens in the G2 phase is, so you are at number 5. So from here it goes back again to number 1. So this is a cell cycle, and that's why it's called a cycle. So you start with the M phase where nucleus division or mitosis occurs, followed by the interphase where the daughter cells are formed from cell division, and it enters the growth phase. The growth phase has the high rate of biosynthesis, G1, then the S phase, which is the DNA synthesis, and then the G2 again, which is the initiation of the mitosis, and it goes back to the M phase. So this is the entire cell cycle. Now, the nomenclature that we are going to use for much of today and in the next few lectures is Rx, would be the rate of cell growth. So why this nomenclature? Because what we are essentially trying to do is we are trying to quantify the rate of cell growth. Now, what happens if you're doing an experiment in a lab, for example? You do a process of what is known as incubation of cells, right? So you take a petri dish or a chemostat or something like that, and you start off with what is known as a starter. So you start off with some cells, and then you incubate them. You provide nourishment, nutrients to the cell. You retain a certain level of carbon dioxide and oxygen supply, and then you maintain the pH at a level at which the cells can grow, and the temperature. So the required elements are a certain temperature. So say up to 25, 30, at most 35 degree, 37 degree at most. So that kind of temperature, you cannot increase the temperature to say 40, 42, the cells are going to die. So each cell, depending on what cell you are trying to grow, would have a temperature range, just like we had in the case of enzyme. If you remember, the enzyme was functional within a temperature range and a pH range. Similarly, so here, for example, a cell would be, for every cell there is a temperature range within which the cell is functional. Okay, so the temperature range could be say 25 to 37 or 40 and so 38, so on, not more than that. So, and then there will be a pH range of say 6.7 to 7.2 or 6.8 to 7.2 or 7 to 7.4. There will be a pH range where it would be effective. Then you would have to maintain a carbon dioxide concentration, say 5% or something like that, a certain oxygen concentration, and then you have to give nutrients so that all the elements that are needed for the cell to grow would be there. So what are all the elements that are needed for the cell to grow? The same elements that are needed for a human body to grow, because the cells are what constitutes the human body. So what could be the elements that are required for the cell to grow? Oxygen I already mentioned. Okay, carbon, one of the essential ingredients. Nitrogen, hydrogen, right? So essentially these are the nitrogen, hydrogen, carbon, oxygen. So these are the essential elements that you require. So you have to provide those elements in terms of nutrients. Oxygen supply is of course there, and carbon dioxide supply is of course there. In addition, you need to provide in terms of nutrients, like for example, what would be the most common nutrient that you provide to the cell? What would be the most common? For example, if you're dehydrated, what is the thing that the doctor or everybody tells you to take? Yeah, what is that? What does it contain? What does it contain? Not saline water. Glucose, essentially, yes. So water containing sugar and salt. So so what would happen is that, what are the elements in glucose? Essentially carbon, hydrogen, oxygen. Okay, so that's the reason that you ask them, because your essential nutrients are here. So you can provide some kind of nutrients like that, and you need nitrogen also. So some ammonia or some other constituent which has nitrogen in it, you provide these. So then what do you do? What is the process of cell growth? Has any of you have done cell growth in the lab? So the process of cell growth is essentially, at home you're excuse me. Yeah, there's a process of cell growth that is used at home. Your mother does it probably, and what is it? Can you tell me what that process of cell growth is? For curd, yeah, for curd, for yogurt. So producing yogurt. So what you do? You take a little. It's the same process in the experiments also in the lab. You take a little bit of starter, right? So what does starter contain? That starter would contain a little bit of yeast. Okay, a little bit of bacteria that is necessary, and then you provide. What is the process? If you think from an engineering point of view, the process of say curd formation, yogurt formation. What is the process? You will take a little bit of starter, which contains the microorganisms that you need to grow, and then you provide suitable environment, suitable temperature, suitable acid conditions, suitable pH, enough oxygen and nutrients and water and so on, so that the thing grows, and the microorganism grows, and within a period of time you will see that the entire milk has been curdled, right? So essentially it's the growth of the microorganism, and what is the time scale for that process? Say around 24 hours. A curdling process would be around 24 hours. So similarly, is the exact same procedure that we do in our homes every day is the procedure that is transported to the labs when you incubate cells. It's maybe a slightly longer procedure. So why do this? Because some of these microorganisms, for example yeast or some other microorganisms that you use may be expensive. You don't, if you want to buy them all the time, it's pretty expensive. So what is done is you grow these microorganisms in the lab. Okay. So for example, one of the microorganisms that is yeast, of course, and things like macrophages. Okay, I talked about macrophages earlier in different point of time. So these are immune cells, and you want to study, for example, the properties of these. So what I'm trying to say is that there are two aspects. One is microorganisms that are grown, that are beneficial for human, scientific whatever activity or whatever, and you want to grow those microorganisms for your use. That's the utilization purpose, and that is something that you can do in lab. That is something that you can do in these factories and so on, and you can do at home also. But then there are other kinds of microorganisms which you don't necessarily need to utilize, but you need them for scientific inquiry. Okay. So for one of them is, for example, immune cells. So if you want to buy these immune cells, monocytes, macrophages from the lab, they're pretty expensive. So you buy some of them, and then you regrow this over a period of time, so that you have a steady supply of these during your experiments. So if you're performing your experiments every day, so I'm now looking at it from a very local lab scale point of view. So if you're performing your experiments every day, you need a steady supply of, say, 10 million or 20 million or 30 million macrophages. So what do? You put a little starter of, say, a million or a couple of millions in a Petri dish, and then you allow the suitable environment, which is that, so you put it inside an incubator. So you have these incubators, many of us have in the lab. So you put it into an incubator, and you allow, so some of these incubators will allow a certain carbon dioxide flow. So we allow a certain carbon dioxide flow of 5% or something, and oxygen flow is of course there, and then you put the nutrients in there. Okay. So this is a process. I'm trying to describe the cell growth process before we go into modeling and more complicated things, because it's important to understand that what really happens. So you put in nutrients, okay, whatever is the required nutrient for the cell, which should include carbon, hydrogen, oxygen, nitrogen, and so on, and allow it to be there for, say, around 24 hours, the same time scale that you would use for a curdling process. Now, after you come back after 24 hours, and you started with, say, 5 million cells, you would expect more or less a doubling. Say, if the doubling time is, we will do that. What is the doubling time? You need to figure out what is the doubling time for the cell. So if your doubling time is around say 24 hours, so after 24 hours you would expect to have 10 million cells. But what do, do you just take the Petri dish and expect to have 10 million cells and start working with 10 million cells? No, you don't do that. You take out a sample out of that little bit, out of that liquid that you have, okay, and then you spin it, okay, in a centrifuge. You spin it in a centrifuge. Why spin it in a centrifuge is to get rid of everything else that is around it and isolate the cells. Then you take those cells below under a microscope. Why do you take those cells under a microscope? Can anybody intuit? So you just took a sample. You didn't take the whole thing. You in a Petri dish or in a beaker or something, you grew these cells. So you started with 5 million cells. You expect 10 million cells to be there. You provided the right nutrients, the right temperature, the right pH, the right carbon dioxide and oxygen flow, and everything was fine. And you expected after 24 hours to have 10 million cells. But what will you do? Will you just take the thing and assume that there are 10 million cells? No, you're not going to do that. So what are you trying to test? What am I trying to test here? Whether there are 10 million cells or not, right? That is what I'm trying to test. And what are the possibilities? Because this is very related to what we're going to do just now. What are the possibilities for? Let's talk about that. So one of the possibilities is that the cell didn't grow at all, right? The second possibility is that the cell grew, but the cell didn't grow at the rate that I thought it would. But then what could be the reason for that possibility? The reason could be that I didn't provide the right environment, the right nutrient, the right temperature, the right pH. Now, I might have provided, then you might argue that, well, I provided all the right environment, then why is the cell not growing, or why shouldn't the cell grow? So if you provided all the right ingredients, then what is the third possibility that is there? So first possibility, I said the cells didn't grow. That would happen only if you went grossly wrong. So if your, say, pH range was 7 to 7.4 for the cell growth, and you put your pH at 6.8, then no cell growth. So that is something that will happen if you go grossly wrong. The second is the cell growth is reduced, which means that the growth kinetics is retarded, is decelerated. There may be reasons for that. Again, pH, temperature can be effects, but small variation. So if your pH range was, say, 7.7 to 7.4, and you put your pH at 6.9 or 6.95, then the cell growth would happen, but it will be slightly retarded. But what else could happen? What could be the third possibility? So the cell grew, the third possibility could be cell grew, but something else happened. Cell grew at the rate that it was supposed to grow. Because see, the calculations typically are not wrong, that if a doubling time has been ascertained for a particular kind of cell, it has been ascertained through a series of experiments, and a specific set of conditions have been specified, right? Particular set of conditions have been specified. Now, if you maintain those conditions, there is no reason for the cells not to double. What is the other possibility? Just think about that. If the cells grew at the rate that it was supposed to grow, what else is a possibility? The third possibility is that cells are dying. It's a very simple, possibility. Cells are growing at the rate that they are supposed to grow, but after growing, that they are dying. Okay, and what could be one of the reasons? One of the reasons could be population explosion. That is, what I mean by population explosion is that the amount of nutrients that you provided was not enough for 10 million cells. Okay, that's a very important point. That you thought that the amount of nutrient that you provided was all right for 10 million cells, but the amount of nutrient that was provided was not enough for 10 million cells. And the same the biological principles that apply for different animals or whatever applies for cells also, because it is the survival of the fittest. And so these cells will start to fight with each other if there is less amount of nutrient that is present. So these cells will compete with each other for nutrients, and the cells that will be depleted of nutrients will die. Right? Is it clear? So this is a very important thing that you have to understand, that just as there is growth of cells or birth of cells, there is death of cells. Okay, and this is a thing that is there, inherently there. So this is a thing that you cannot get rid of. That you cannot assume that you put in nutrients and everything, and the cells will grow as it is. So one of the possibilities of cell death could be that there's not enough nutrient for that population. The second possibility is that there is defects in some of these cells that are growing, and as a result of which they are dying. So what am I supposed to do now? That's why I take out a sample from there and centrifuge it. Okay, and first of all I drain the liquid around it, centrifuge it, and then I put it under the microscope. What do I do under a microscope? I try to measure the amount of dead cells, that is there. Okay, so how do I measure it? Let me show you. So what you do is you take your cells. Now these are you take a dish like this. Okay, there are these dishes that are available in the lab, flat dishes with markers on them. Okay, so what you do is, you put that sample, after you have centrifuged it, you put that sample on here, and by the way, what you have to do is, after you have centrifuged it, you dye it. Okay, you dye the cells. And so what happens is that when you put the cells in like this, these cells will be, so these dots that I put over here, can you see the dots? So the dots that I put over here are the cells. Now the cells, when you dye a cell with a certain dye, cells that are living would be of a certain color, and the cells that are not living would be of another color. Okay, because the cells that are living the dye would get diffused into the cytoplasm of the cells, and the cells would have a certain color. So if the living cells are red, then the other cells could be blue or some other thing. So now we put this under a microscope over here. Okay, and then you look at the cells, so you can figure out. So you have a count of, so these blocks are there so that it's easier to count. Okay, so you have a count of the, first you have to count the total number of cells, and then you have to count the number of living cells or the number of dead cells, whichever. People typically count the dead cells. So the total number of cells and the number of dead cells. So then you will have an average fraction, say n equals number of, n average is number of dead cells over number of living cells. Okay, so you will have an average for the sample. Now assuming that this is a random sample, you need to do this a few times. You just cannot do it once and trust that. You need to do it 3 or 4 times, and based on these n, basically let's call this n then. So n average would be, so ni, I going from say 1 to m, if you have done this m times, over m. Okay, so that would be your average fraction of dead cells in the system. So from that you can just multiply by, if you are supposed to have like 10 million cells or something, then you just multiply that, what would be my total number of living cells. Now, how do I know whether actually I have 10 million cells, right? That is the question. So, I expect 10 million cells, does not necessarily mean that I will have 10 million cells. So, how do I know that? What I do is I take, say for example, if my total liquid is like 100 milliliter or 50 milliliter, say I take a 5 milliliter sample, okay. And now what I have done over here is I have counted, if you look at this, I have counted the number of cells, not just the number of dead cells, I have also counted the total number of cells in the system, right. So, you can just multiply, if my 5 milliliter gives me 1 whatever, you know, or 1 milliliter, I take a 1 milliliter sample out of that. So, 1 milliliter gives me this amount of cells, then 50 or something would be your 100 milliliter was supposed to give that. And because this is I am doing this again and again, okay. So, then there is an average thing, that is there is an averaging that is coming out. So, you calculate the n average, which is the fraction of dead cells, actually this should be number of living or number of total cells, okay. And similarly, you calculate the total C total, let us call it C total average of that. So, if you are taking if you did it for 5 times and you calculate the C total for each. So, 1 over m sigma of C total I. So, that would give you the average number of cells within that, and you can multiply that by the number to find what is the total number of cells in your system. So, then what you find, for example, at the end of the day is that while you are supposed to have like 10 million cells, you end up having say 8 million cells or something. And what you do next day is that you take out of those 8 million living cells, whatever you have, you take out whatever you need for the experiments, or say you might need lot more, you might need 50 million for your experiments. So, you use those 50 million cells or say 40 million cells, and you recycle 10 million cells, okay. So, you put those 10 million cells again for next day, put in nutrients and put it in the incubator for 24 more hours and recycle it. Why do you recycle it? Because if you are running your experiment every day, you need a steady supply of cells every day, fine. Now, what do you do with the 8 million cells or whatever 40 million cells that you have? Can you use it? No, you have to wash them, okay. So, you need to wash the cells, because the cells was with the nutrients. Now, if you are going to do your experiment, you need to wash it, and then centrifuge it possible, that is drain away the liquid, and then you can use the cells for your purpose. So, this is the essential process of cell growth in the lab, okay, in the lab while you do your experiments. What we are going to study here in today's lecture and part of today's lecture and next lectures is what is a mechanism, what is a quantitative way you can measure these cell growth. So, these are experimental ways of I talk to you about how you actually go and measure the cell growth under an experiment, but what would be quantitative mechanisms of measuring cell growth, what are there models of cell growth, and what if a much more complex system is involved. For example, you do not have one kind of cell and one kind of nutrients that are growing, but you have multiple kinds of cells that are growing, and same set of nutrients can foster the growth of different cells, right. So, if you are trying to incubate 2 or 3 different cells at the same point of time, then what happens? Are there interactions between these cells? Possible, there could be interactions between these cells. So, some of these complicated things. Why? Because see, when you are doing these experiments in the lab, it is more of a pristine controlled atmosphere, right. But when these things happen in real life, you really do not know what kind of interactions there could be. There could be varied, complicated, intricate interactions between these cells, okay. For example, many of these bacterial diseases that we get, especially in countries like India, tropical countries or subtropical countries, we get this lot of diseases in summer, late summer, and then in the monsoon. Can you intuit why we get all of these summer diseases or things like that more here than say in the colder countries? What kind of environment? Yes, that's right. But yeah, it's a hot, humid, humid climate. So, essentially what is happening is these, for example, these bacteria, many of these bacteria that are responsible for gut infections and other kinds of infections, even respiratory tract infection, a lot of these different kinds of infections that are there. So, what happens in colder climate, because the temperature is very low, these cells cannot grow. So, temperature is a major hindrance in their growth process. Okay. Whereas for us, if you think, the temperature is actually very, very ideal for the cell growth process. This is ideal temperatures are around 25 to 37 degrees centigrade, or 20 to 35, 37 degrees centigrade. And the temperatures that are offered in that are available in these tropical and subtropical countries are actually ideal for that. Okay. So that is one, and because of humidity, now many of the oxygen and oxygen is available everywhere in general, but because of humidity, the moisture that is there in the air is also something that the bacteria can use. Now, why am I talking about this? Because in the human body, in the physiological system, so when you are doing a controlled experiment in a lab with one single cell, it's a straightforward process, and that is something that we are going to study today. But in the later lectures, maybe tomorrow or in the next lecture or the one after that, we'll study what happens when there is a complex dynamics between these cell growth processes that occur. And why are we talking about complex dynamics between the cell growth processes? Because in physiological systems, in human body, that is what happens. You do not have a controlled atmosphere where you are very carefully growing one cell and looking at it under the microscope, right? So that's not possible. So all these cell growths are connected to each other. So the growth of one cell might suppress the growth of another cell. Okay, and the amount of nutrient that is available, for example, E. coli, colitis, E. coli, B. coli, some of these colitis bacteria that are very well known to grow inside the human body. When they grow, they will take away much of the nutrient that is available, right? And they will therefore suppress the growth of other bacteria, beneficial bacteria, that is necessary for example, producing enzymes for digestion. Okay. So, the digestive enzymes are often produced by microorganisms in the system. One of the things that you might know is that, might or might not know, but I'll talk about it. For example, when the doctor, if you have a say upper respiratory tract infection, some of us are getting it these days because of season change and all that stuff. So the doctor gives you some antibiotic, right? For example, azithromycin is a typical antibiotic for upper respiratory tract infection. So doctor gives you that. So when you take those antibiotics, have you ever noticed, if you take those for 3 or 5 or 7 days, that you have bad stomach, right? You have bad stomach. And why is that? Useful bacteria in the guts, exactly. That is the reason. See, because in the guts, human guts, these useful beneficial bacteria that are used for digestion and all gut related, intestine related activity are being constantly formed. Now, the antibiotic is the one that kills bacteria, and most of the antibiotics, not all, but most of the antibiotics do not have the capability to discern or differentiate between a good bacteria and a bad bacteria. So while it kills the bad bacteria that is there in your lung or elsewhere, and these go through the blood, right? So they are everywhere. So even if it wants to kill, so nowadays we have localized antibiotics, but despite that, so because it flows through the blood, so while it is supposed to kill only the bad bacteria in your lung or your upper respiratory tract, it also kills the good bacteria that is there elsewhere. It can kill the macrophages that are there, reduce your immune system. That is a very bad part of it. It can kill bacteria, beneficial bacteria that help in digestion in the gut. Okay, so that is why you have these problems. And what is the antidote? One of the things that either the doctor tells you or gives an additional medicine when you have a bad stomach and you take because of antibiotics. So when you take antibiotics for long, you're bound to have bad stomach. This is something that will happen to you. So what is the antidote? Have you ever thought about it? Somebody ever told you that? You eat yogurt, curd. Why? Because curd regenerates those microorganisms. Okay, and sometimes what happens is that, so these times pharmaceutical companies are getting smarter. So the doctor used to give you, or maybe used to give you with the antibiotic another medicine, but these days you will see, so some of the antibiotics will say with Saccharomyces cerevisiae. Okay, so which means the yeast is there in the antibiotic. So while the antibiotic is killing the yeast, it also has a ready supply of yeast in it to regenerate these antibiotics. Okay, so this is something that is coming up new in the probably in the last couple of years in India. You would have seen that pharmaceutical companies produce these drugs the antibiotics which come with yeast. If it doesn't come with yeast, then the doctor either gives you a separate antibiotic, a separate tablet for generation of yeast, or tells you that you eat a lot of yogurt so that to regenerate the yeast. So that is the kind of system that I'm talking about, where there is complex interactions between bacteria. So one cell is growing at the expense of another cell, maybe. Okay, so or maybe one cell is killed, along with that another cell is killed. So some of these complex interactions we'll study, and we'll try and see that how this could be extended to other complicated systems. Okay. So, but let us start with the simplest thing today first, which is the cell growth as I described in an experimental environment with a very simple controlled experiment. So, the nomenclature that we are going to use are written out here. Rx is the rate of cell growth, which is the volumetric rate of increase of cell concentration. Okay. So, just as you had the rate of reaction in an enzyme, this is the rate of cell growth. X is the cell concentration, we will use the notation X for it, usually dry cell weight per volume. This is how it is defined. Okay. Dry cell weight, that is, you drain away the liquid, just as I said, you drain away the liquid, centrifuge it and take the dry cell, and dry cell weight per liquid per volume, and mu is the specific rate, that is growth of cell per unit concentration of cell. Okay. Cell growth per unit concentration of cell. So, these are the nomenclature that we are going to use, and you may want to note them down. So, Rx is the rate of cell growth, volumetric, X is a cell concentration, dry cell weight per unit volume, and mu is a specific cell rate of growth, that is Rx per unit concentration. Okay. So, one of the reasons we are studying this cell growth process in this lecture is, of course, that this is a reactive process. Okay. So, what happens, what is the reaction that occurs during the cell growth, that we need to understand. So, as I told you that what are the nutrients, what are the components that are necessary? Nitrogen, hydrogen, carbon, oxygen, right. And one of the main sources that we give it through is glucose. Glucose, of course, does not contain the nitrogen source, so we need to add in a nitrogen source. So, the nitrogen source that we add in is ammonia over here, as it is written, and this is a kind of reaction that happens. Okay, let us look at this. So, first the one that you have here, alpha CHLOM, this is a, let us call this a glucose source, okay, the carbon source, it could be glucose or some form of carbon. Then you have the ammonia, which is the nitrogen source. Oxygen is plentiful, any time you do a cell incubation, oxygen is there. In addition, what has not been described here, you can give a little bit of carbon dioxide, because the incubator that you use typically has a possibility that you can use 2%, 3%, 5% carbon dioxide, and people give that often, because that acts as an additional source of carbon. But you may not also, but because carbon is there in this carbon source that you give, glucose or whatever. So, so then what happens is that this carbon source changes itself, and you get from that carbon source some other form. So, because a part of it is being utilized. So, this is the C is the elemental composition of the cell that is going in, and sorry, yeah, this is the source that it changes itself, but this is the elemental composition of the cell, CHAOBNC. Okay. So, this is not the cell itself, but the constituents of all these chemicals inside the cell. So, we assume that carbon, hydrogen, oxygen, nitrogen are present in the ratio of 1:A:B:C. That is the assumption here. Okay. And then this is what is left, and then water is left, and carbon dioxide is left. These are very straightforward things, but this is what we are more interested in. Okay. And this is, as I said, this is what is left. So, you start with this and this. So, you add a carbon source. So, essentially to remember that you use a glucose or some kind of carbon source, nitrogen in terms of ammonia, say here, and oxygen, and this is the one that is important to us. This is what you get inside the cell, and this is what is outside all of it. Okay, fine. So, what happens in the cell growth process? So, in the cell growth process, ATP is generated. Okay, and this is something we discussed before. ATP is adenosine triphosphate, and I think you had done this in Professor Neogi would have talked about this a lot. So, it is essentially a energy currency of the cell. Okay. So, when we are talking of matter, okay, the cell growth process, the nutrients, what we are essentially interested in is energy, right? That all of us is essentially interested in it. Starting from the human body to the human civilization, everybody is interested essentially in energy. So, but what is the process? The process is you take in matter, you burn the matter to generate energy, right? So, ATP is known as the energy currency of the cell. So, it is like money, it is like banknotes. How much money you have, you measure in terms of banknotes. The cell measures how much energy it has in terms of the amount of ATP it has, and it all doesn't spend all the energy together. So it converts from ATP, adenosine triphosphate, to adenosine diphosphate, and adenosine diphosphate to AMP, adenosine monophosphate, and it is a miser thing. So it kind of spends the energy money little slowly. So ATP is the energy currency of the cell. NADPH is it is part of the cellular electron transport mechanism, NADPH, NADPH plus. So essentially this is a very important thing that, and we have done this in some of the course before, so I don't want to repeat that. So many of the cellular transport processes, if you remember from what we did in another course before, are governed by NADPH, and because of the cellular electron transport that is there, and this is based on the oxidation of the carbon source. So apart from the nutrients, so there are three outputs that are there in the cell. So this is the one, C, H, A, O, B, and C. Okay, this is the material output. So the cell retains this material, but what the cell is essentially interested in is energy, and that energy is stored in terms of ATP. So there will be a material or a bulk increase of the size of a cell. So I was explaining the cell growth process like the growth of a baby after it's born. So initially, if I am to go back here, so initially this is a cell cycle. So initially the cells are born through mitosis and daughter cells are formed. So you start with the M phase, the mitosis phase, then the interphase when the daughter cells are born, and then this is the phase that we are looking at, high rate of biosynthesis, the G1 phase. Okay, so the M phase is the nuclear cell division, mitosis, formation of the daughter cells. Now this is like a birth of a baby, and then when the baby is born, there is a high rate of biosynthesis that goes on for years together, maybe 15 or 16 or 18 years, till the baby turns into an adult. So there is a what is happening? So two things are happening in this high rate of biosynthesis. One is the material increase of the volume of the cell, just like a baby grows into a mature adult and the size of the baby grows, and also the energy that is used and stored by the cell increases, that is the ATP of the system increases. If it stores more energy, it can use more energy, right? So then it's the DNA synthesis phase that we are not looking at, and the DNA synthesis occurs until the DNA content of the cell has doubled, and then again it goes back to phase 1 over here, initiation of mitosis. So it's a cyclical process. So what we are looking at here is the phase 3, the G1 phase rate of cell biosynthesis. So this is an interesting thing. So this is an experiment that had been done in a batch reactor. This the experiment that I described to you just now at the beginning of this lecture, that is done in a lab scale. So what really, if I am monitoring, if I am taking out samples from time to time, okay, what I talked to you about is I start here and I talked about the end product, right? I started here 0 hours, and this is say 24 hours, and this is where I measured the cells, fine, or somewhere around here 24 hours, and I measured the cells. What if I am taking out samples time to time from my petri dish or wherever I am incubating and measuring the concentration of the cell? How do I measure the concentration of the cell? Just as I described, you take it out, centrifuge it, put it on the microscope and measure the concentration of the living cell. Then what do I find? And this is a very interesting thing. So this is what it shows you, cell number versus time, okay, and this time is say in the length scale of hours, okay, so 24 hours or 20 hours or whatever. So initially what you find is, look here, initially what you find is known as lag phase, okay. So initially what you find is you put the cells in and nothing happens for a period of say 1 hour, 2 hours, nothing happens. Because why is it? Because once it, before you go to the phase where it starts to divide, okay, what is the cell growth? Let us understand the cell growth process. So what we are looking at is that the rate of biosynthesis is what I just showed you, the reactions that I showed you, right? But you want the cells to double, right? And that means that it has to go to the initiation of the mitosis phase. So initially, is it clear to everybody? If initially you have the cell growth phase, which is the high rate of biosynthesis phase, and once the high rate of biosynthesis is over, that is, the cell has attained a mature adult stage, okay, then it starts to divide, forming daughter cells. Now what you are measuring over here is the total number of cells in the system, right? So if you want the cells to double or say increase in number, then first it has to go through the high rate of biosynthesis phase and generate all the energy that is required, and then it starts to divide. So initial lag phase is when the only biosynthesis is happening and there is no cell division, okay. And then after the cell division gets over, then you start the growth phase, that is, growth means cell growth phase, cell number growth phase, growth of cell numbers, or in other words, the mitosis of the cell division occurs. Is it clear? Okay. So, this turns out to be an exponential phase. So, this growth is an exponential phase. Okay. So, growth is occurring. Then you have a stationary phase, that is the cell number reaches, so cell number is constant initially, then it increases very fast exponentially, and then it reaches a stationary phase. So, if you would like to have high, and then what happens, it starts to decrease. Now, if you had like to have maximum output from your culture, okay, so the culture process that I described, what would you do? You would stop somewhere here, right? You stop somewhere here and take the cells out, fine. So, that is why it is important for us to figure out what is the dynamics of the process, because if I know the dynamics of a certain process, so if I know certain cell A or B has a certain dynamics, then I would stop my culture at, say, I would know, okay, after 16 hours the stationary phase starts. So, I stop at 16 hours, I stop my incubation of culture at 16 hours, take it out. Now, you have to tell me now, I told you that this is the growth that is happening, you have to tell me that how the stationary phase is attained, what happens in the stationary phase. Why would it stop? Would the cell growth stop, and if so, why? What is that? No, nutrient is not deficient in this phase.
Cell division stop.
What is that?
Cell division stop.
No, cell division doesn't stop. If nutrient is not deficient, why would cell division stop? Cell division will stop only if nutrient is deficient. Okay. Why is it stationary? Why is the cell number not growing any further? Right, correct. The growth rate and the death rate are equal in the stationary phase. Okay, it's not that nutrients are deficient. If nutrients are deficient, then cell division wouldn't occur. The growth rate and the death rate are the same in this phase. So, if you look here, what's happening is that that's why it's a stationary phase. Then comes the death phase, where what you said is correct, that the nutrient is deficient in the death phase, and then the cells start to fight for each other. As a result, not that cell growth or cell division completely stops, but the death rate is higher than the growth rate. As a result, the death phase is there. Okay. So, this is the total process. So, now if I want to model the dynamics of the cell growth process, I have to find a way of incorporating all these different phenomena, the lag phase, the growth phase, the stationary phase, and the death phase. Okay. So, these 4 phases. So, what we will try to do is try and see what kind of model we can develop, or what kind of models have been developed over the last 50 years to try and model this. So, we will go slowly, let us understand one by one. So, the first thing that we look at is the exponential phase, the exponential growth phase, which is the easiest to model. Now, this exponential growth phase is given as dx dt equals mu x. Okay, you might want to make a note of this. So, dx dt equals mu x. What is mu? Mu is the specific growth rate of cells. Okay. So, now we are just modeling only the exponential growth rate part, not the other 3 phases, only the exponential growth phase. So, the dx dt equals mu x. Mu is the specific growth rate, it is like a reaction rate constant, okay, and x is a cell concentration. So, what this means is that more is the number of cells you start with, the more is the possibility that you will generate cells. Why is that? Because every cell would give rise to 2 cells through mitosis at each point of time. So, if you start with 5 million cells, you expect to have 10 million. If you started with 10, you expect to have 20. Fine. So, that's why it's linearly dependent, and you can integrate it like we have done over here before here. So, you after integration you get x equals x naught e to the power mu t minus t lag. T lag, why? Because you assume that up to the time t equals t lag there is no cell growth. So, x equals x naught, what is the initial concentration of the cell that you have started. So, that is your starter, you know, when you are doing the curd, making the curd or the yogurt. So, the amount of cell microorganisms or yeast that you put in at the starting, that is your x naught, fine. So, that is at t equals t lag. So, up to t equals t lag there is no increase in the cell number. So, x equals x naught e to the power mu t minus t lag, and once you integrate it you get this. Now, what we are interested in is the doubling time. Okay, what we are interested in is the doubling time. It is like a half time, you know, in radioactive decays you have half time, and here because it is a growth process and not a decay process, you have something called a doubling time, and that is what we are interested in. So, Td is the doubling time and is given as ln 2 over mu, mu being the specific growth rate of cells. Okay. So, mu, if you go back to reaction, just like your reaction engineering where you had K as defined as 1 over x del t, here you have mu as defined as 1 over x del t, x being the cell concentration. Okay. So, this is we are at the exponential phase. Now, what we look at is that look at how to evaluate this mu. Is this mu a constant? No, it is not a constant. Is it something like a reaction first order reaction rate constant? No, I'm afraid it's not. Then what does it depend? Because it depends on a number of factors. So, what does it depend upon, and how do we obtain a model for this? So, there are several models for evaluating this mu, the specific growth rate, and I'll go through these models one by one, and I may not finish in this lecture, but let's see. So, the first model that we look at is known as a Malthusian model, M-A-L-T-H-U-S-I-N, Malthusian model. So, this model assumes that Rx is mu times x del x del t, and x naught is given as e to the power mu t minus t lag, and this model assumes that mu is a constant. What is the shortcoming of this model? If mu is a constant, then what happens is the exponential growth phase continues forever. There is no way to predict the stationary or the decay phase over here. Okay. The only way is to be able to use this model and cut it off at a certain time scale. The next model is known as the logistic model, and to overcome this shortcoming of the Malthusian model that was predicted. So, you see these are very old models actually. These people Wellhurst and Perlin Reed in 1844 and 1920 proposed the addition of the cell concentration as a dependent second term, which means that the reaction is no longer first order in x. Okay, that is what they said. So, that was an intuitive idea. What was the idea? That let's go back here to the picture, and that will. So, they said, well, it's not, it's decreasing after a certain value of the x cell number. So, why not make it a part of it to be dependent on cell number, but inversely. So, as cell number increases, then your growth rate decreases. So, this was the intuitive thing. They were doing this experiment, and they are figuring out that after a certain cell number it starts to decrease, and then they thought, why not make it like such that initially it is dependent on cell number, but after a certain cell number it starts to decrease with that cell number. It is not a bad idea actually if you think of it. It may not work, but it is not a bad idea. So, it is like this. So, you know, initially for small values of x, then this number it does not kick in. Okay, the quadratic part does not kick in, it goes almost linearly, and then when x reaches a certain value, this number becomes larger, and therefore, the effect of quadratic effect, because of negative quadratic effect, it starts to decrease. So, this if you integrate, and you can still integrate it by the method of what? Not separation of variable, partial fractions. Yeah. So, at x equals x naught, t equals 0 at x equals x naught, and actually you can remove this t equals 0 and you can put t equals t lag if you want to. That way you can include the lag phase also, and when you integrate it, this is what you get: x naught e to the power kt 1 minus beta x naught 1 minus e to the power kt. Okay. One of the things you can do is, I do not think I have that, is you can go and plot this in the computer using some numbers of k and try to see that whether that predicts the experimental. So, what I showed you, the plot I showed you is the experimental plot, and whatever model we come up with has to kind of predict that experimental plot properly. Okay. Is that right? Shall we go on? Okay. The next model is the most important model that we are going to do, and this is something that you need to remember, is known as the monod model. Okay. This was founded by J. Monod, and it was in 1950 in this paper, Annals of Institutes of Pasteur. 1950 he founded this model, and the reason it is the most important model is because it is the most successful one, and almost all growth processes that are there in the human body today are modeled by the monod model, or what is known as the modified monod model, and I will teach them right one after another today. So, the monod model says that, and it is an important thing. See, what monod model says is that no, the x itself, so this thing is right, Rx is mu times x, and you do not need to include another part, but let this mu be dependent on the substrate. Okay, that is what. So, if there is a depletion of the substrate, what happens is the rate constant for growth decreases. Okay. And if you look at this interestingly, what have they done with the rate constant? They have put a Michaelis-Menten kinetics there in the rate constant. Okay. So, now my dx dt according to the monod model as a result okay. So, this is my model. So, the model is still first order in x, but a Michaelis-Menten in substrate concentration or in terms of nutrient concentration. Okay. Is it clear? So, what this means is that if you look at dx dt versus s, then it would have a thing like this. Okay. So, it grows as it increases, but beyond a certain point, beyond a certain point in s, it saturates out. Okay. So, this is a Michaelis-Menten versus x, it increases first order as we had done before, and then it saturates out to a 0th order, which ensures that the rate constant itself decreases. Okay. But still it does not include the death phase, remember, or the stationary phase, but it ensures some kind of saturation beyond it, does not grow unlimited. Fine. This is the monod model. Is that clear with everybody? And this is something that you need to remember, by the way, what I gave you today. This what I wrote over here on the sheet. Yeah, this formula dx dt for this is for monod model. This is what you need to remember, and you also need to remember the modified monod model, because that is also something that is used. So, two forms of the monod model. The rest are okay. You do not necessarily need to remember for your for everything, but well, for your test, of course, you need to remember. So, dx dt for the monod model is mu x times mu max Sx over Ks plus S, and mu is the Michaelis-Menten kinetics. It goes to first order, that is mu max S over Ks for S very, very small, and goes to a constant mu max for S very large. Please make sure you make a note of this. Done? Okay. So, this is a monod model, and what I want to do next, as probably the last thing today, is a modified monod model. So, the modified monod model is a slight modification on the monod model. It is found experimentally that the rate of cell growth decreases at high values of initial substrate concentration S naught. So, it was a strange thing that was seen, that if you increase the substrate concentration is good, okay, but if you increase it to a very high value, then that, instead of helping the cell growth process, was retarding the cell growth process. And how do we include that? So, that was included in mu, and what was done is a simple thing. So, monod model had mu max S over Ks plus S, and the modified monod model simply put this Ks S naught. So, if the substrate concentration is very high, S naught say, then decreases the mu itself decreases, because you put it in the denominator, and as a result it would have a decreasing effect. The rest is fine, that is the mu max, that is dx dt, is still equal mu times x, where mu instead of mu max S over Ks plus S is given as mu max S over Ks plus S plus Ks naught. So, this is a modified monod model, and it sometimes works because of so too much nutrient is not good either, too much of a substrate concentration is not good either. Okay. So, let us finish this, the Konak model, and so this was founded a little later than a monod model, and this is how it is, and it's best that you make a note of this as well. So, in the Konak model, again the major in all these models, except for the second one that we did here, just a sec, except for the logistic model, all the models follow this Rx equals mu x. Okay, this should be very clear in your head, that all the models follow Rx equals mu x, and all we are doing, except for this logistic model, all you are doing is trying to figure out how mu is. So, the monod model gives mu as mu max S over Ks plus S, the modified monod model gives mu max S over Ks plus S, Ks S naught plus S, and the Konak model gives mu as the differential equation itself. So, what this says is that mu is a function of time, the specific growth rate itself is a function of time, and that equation is given as, so the model stays the same, but mu is a function of time also. Okay. So, mu is not a constant anymore, it is not necessarily dependent on concentration also, substrate concentration also, it is a function of time which you need to integrate. So, when you are integrating dx dt, you need to integrate it in coupled with the mu equation, right. So, dx dt equals something and d mu dt equals something. So, you need to integrate these two equations together. So, this equation is given as d mu dt equals k times mu max minus mu to the p, where p and k are adjustable variables. Now, for p equals 1, mu max becomes linear. So, mu equals mu max e to the power minus k s, okay. And this becomes mu for p equals 1, and for p not equals 1, this is what you get. Actually, I think I am sorry, this is a typo over here. It should be del mu del s, not del mu del t over here. This should be del mu del s, the Konak model. Just make that change. So, this is del mu del s equals k times mu max minus p mu to the power p. So, for p equals 1, you get mu equals mu max 1 minus e to the k s. For p equals 2, this is what you get. P not equals 1, this is what you get, and for p not equals 1, the formula that you get, if you put p equals 2, then you get the monod model, get back to the monod model. Is it clear? So, this is slightly complicated, so let me run through this, and there is a typo out here also. So, this is del mu del s equals k times mu max minus mu to the p, okay. And so, let me write, since there is a typo, I will just write it. So, del mu del s equals k times mu max minus mu to the p. For p equals 1, you get mu equals mu max 1 minus e to the k s. P equals p not equals 1, you get mu max 1 minus p minus mu max minus mu into 1 minus p equals 1 minus p k s, and for p equals 2, you get mu equals mu max s, mu max over k plus s. This is the monod model, okay. So, what we need to do is we need to kind of remember the monod model, the modified monod model, and the Konak model, which is a generalized form of these, and how you get the monod model from here. So, we will continue in the next lecture. Let us stop here, and okay, thank you.